Good morning, students. In this video, we'll be going for the structure of DNA and we'll be emphasizing mainly nucleoside, nucleotide and the formation of polynucleotide. Dear students, when we speak about the structure of DNA, to know the structure, we need to know the ingredients to form that structure. First thing comes, the ingredient is the pentose sugar. Pentose sugar, and pentose sugar is having like this structure: 1, 2, 3, 4, 5 and okay. Second most ingredient that comes here is the nitrogenous base. In case of DNA, the nitrogenous base are adenine, guanine, cytosine, and what? Thymine. These four makes the nitrogenous base of the DNA. Pentose sugar means the deoxyribose sugar in case of DNA. Third most important ingredient is the phosphoric, PHOSPHORIC, phosphoric acid. Okay, it is denoted by P. Formula is H3PO4. So these are the ingredients which makes the structure of DNA. So step by step, let's know how the nucleoside is formed. Nucleotide, then polynucleotide. So we'll be going for nucleoside first. Okay. This is a pentose sugar. In case of DNA, it is called deoxyribose sugar. When a pentose sugar unites with a nitrogenous base, this nitrogenous base, NB, nitrogenous base, any one of the nitrogenous base. What are the any of the nitrogenous base? It can be adenine, it can be guanine, it can be cytosine, it can be what? Thymine in case of DNA. Which one is T? Which one is replaced with that of the mRNA nitrogenous base or the RNase nitrogenous base? Now, what happens in this case? This is sugar, pentose sugar, uniting with any one of the nitrogenous base. It may be adenine, it may be what? Guanine, it may be cytosine, it may be thymine. Then we call it to be nucleoside. Understood the thing? Nucleoside. So this is nucleoside. Done. Now come to nucleotide. What is that nucleotide? Nucleoside plus phosphoric acid. I am writing here. See, nucleoside plus phosphoric acid, H3PO4. It produces what? Nucleotide. It forms nucleotide. How it forms? Let's have the idea. See, this is the pentose sugar, deoxyribose sugar in case of DNA, ribose sugar in case of RNA. Here you are finding the nitrogenous base, This is nitrogenous base. Okay. This is nucleoside. Any one of the nitrogenous. I told you it may be adenine, it may be guanine, it may be cytosine, it may be thymine. When this nucleoside unite with phosphoric acid, it is denoted by what? P. It is denoted by P. That means nucleoside plus phosphoric acid. It is called what? Nucleotide. Understood the thing? So what is nucleotide? When phosphoric acid unite with the nucleoside, then it becomes what? Nucleotide. Now we'll be going for the last one, that is called the polynucleotide. Polynucleotide means what? Simple thing. Nucleotide plus nucleotide. It is called what? Polynucleotide. Okay. Nucleotide plus nucleotide, it forms what? Polynucleotide. But how it forms? Let's have an idea about it. Here we have done it. This is pentose sugar. This is nitrogenous base. As I told you, any one of the nitrogenous base may be there in case of DNA, adenine, or it may be guanine, or it may be thymine, or it may be cytosine. Any one of the nitrogenous base, when it is uniting with the sugar nucleoside, then we found phosphoric acid uniting, and it forms what? Nucleotide. Now see another thing. We are going for polynucleotide now. How it will be formed? That means two nucleotides need to be united. So here also another nucleotide I have drawn. Okay. Here there is the occurrence of nitrogenous base. Can you get it? So again the same thing. Nitrogenous base can be only one. It may be adenine, it may be guanine, it may be cytosine, it may be thymine. This is deoxyribose sugar. And here also you are finding phosphorus. So this phosphorus is uniting with that of the two nucleotide. That means how it unites? I am coming to that. So union of that nucleotide with that of another nucleotide forms the polynucleotide. Now last part, how it unites. How that is united. Okay, we'll be going for that. The last part we'll be going for. Next one is called phosphodiester. Phosphodiester. Phosphodiester linkage. What is that phosphodiester linkage? Okay. Now see, try to get it. I am rubbing this part as we have done all this part. Completed. Phosphoric acid is having this formula. It is having COH. Oh, H3, 3HP1 and O4, H3PO4. So what happens in this case? OH of phosphoric acid. Understood? Forms a bond with the OH of the sugar. Understood the thing? Now what happens? In this bond, when it forms, it gives out one molecule of water. One molecule of water it gives out. And that bond which it forms, it is called CO bond. Got it? And that is glycosidase bond. Okay? And it is called ester linkage. It is called what? Ester linkage. Understood? Now see how that happens. Here. So, this is sugar. This is nitrogenous base. This is phosphorus. This is another sugar. This is nitrogenous base. This is the phosphorus which unite. This phosphorus has OH. Try to get it. It makes a bond with the OH of the sugar here. Here also it makes a bond. Here another OH is there. Makes a bond with the OH of the sugar. Understood? So that bond, it is CO. Here also this bond is what? CO. Got it? Is called ester linkage. This is called what? Ester linkage. How many ester linkage? Phosphoric acid. One phosphoric acid is forming with that of the sugar. It is forming two. One here, another here. So two. That is called diester linkage. Diester linkage. So because of this diester linkage, polynucleotides are formed. Understood? So what we have discussed in this part, it is not completed. I will be going for the rest part. What we have discussed? We have discussed the units of DNA. That is the pentose sugar. Then we have discussed about nitrogenous base, phosphoric acid. Then we have found how the nucleoside is formed. Then nucleotide. Then finally polynucleotide with the help of the phosphodiester linkage. Thank you, students.